Hello, my name's Niels. Welcome to the PCSIG webinar. Really excited to bring you a couple of speakers today from the University of Bologna. We have Laura Mercolini, sorry Laura, I mispronounced your name, who's the Associate Professor of Medicinal Chemistry at the University of Bologna, and Michele Protti, who's a Senior Assistant Professor of Medicinal Chemistry at the University of Bologna. And they're going to be talking to us today about microsampling for illicit drug testing. If you have questions as we go through the presentation, please submit them through the chat function. We'll also be recording this event, and it will be available through the PCSIG website in a few days. So with no further ado, I'd like to hand over to Laura.
Yeah, thank you. Thank you very much, Neil, for this kind introduction. And hello, everyone. And thank you very much for being here attending this seminar. I'm glad to be here together with my colleague Michele Protti from University of Bologna, the so-called Alma Mater Studiorum in Italy. We are from the Department of Pharmacy and Biotechnology, and we are working in the field of microsampling and patient-centric studies since some years, actually since a long time. And so it's our pleasure today to talk about some applications and method development and validation in the field of microsampling. In particular, my part is referred to microsampling for illicit drug testing. And I will give you an overview of some methods and technologies that we are exploiting since this last more than 15 years, actually, and some analysis that we have performed in this field. I am a medicinal chemist, and my expertise field in particular is the one of pharmaceutical analysis and pharmaco-toxicological analysis and forensic analysis. That's why I'm engaged in the analysis of some drugs of abuse, some illicit compounds that can be simply illicit compounds and that's it, or regular drugs, prescription drugs that can be misused. Okay, I would like to start mentioning the fact that in each analytical process, the sampling procedure, the very first step of an analytical process is the sampling itself. In particular, in our analysis, we apply the processing to biological samples, so plasma, urine, blood, saliva, oral fluid, tears, sweat, and so on. So all biological samples that we can have in our mind. But anyway, even if the analysis is applied to other non-biological samples, the point is that the sampling itself, the step of sampling itself, is very important. And reliable analysis needs to start from careful sampling procedures. Absolutely, this is the first line of my slides, of this very first slide, because actually it is not an easy task. And in fact, there are several challenges that can be related to the sampling step of each analysis. And the ones that I have reported here are simply a selection of all the challenges that can affect sampling. And in particular, these challenges can represent problems, issues, absolutely for the whole analytical and bioanalytical process. And I think that in this moment, drug analysis, forensic analysis represent the current, the state of the art. And at the same time, they should be considered modern analysis. I'm mentioning that because on the contrary, several times you can find in the scientific literature even very new, updated analytical papers about drug analysis and forensic analysis and drugs of abuse analysis that actually use very classic analytical techniques also in terms of sampling. And in my opinion, the modern analysis nowadays should be, for example, a sustainable analysis, an analysis that really cares about ethics, an analysis that can exploit point-of-care testing like home sampling, self-sampling, that can perform centralized sample collection, and also dealing with subject and patient sampling. This modern analysis should be also attentive to the compliance of subject and patient. And in my opinion, there is one word that can put all these four very important aspects together in terms of future perspective, and that are important for the current and the future drug analysis and forensic analysis, and this word is microsampling. I think that through microsampling, it is possible to perform really a modern analysis, and it is possible to perform analytical processes that are able to take into consideration these four elements altogether. But what's microsampling? And I think that in this series of seminars, there are a lot of people in the audience that already know very well microsampling. But this slide is just to present in a very easy way that in this last 15-20 years, we are moving from conventional sampling to microsampling in different fields. And the analysis of illicit compounds is one of these. And so basically, working in the field of microsampling means working with very minute amounts of biological samples that are for sure, in most cases, volumes lower than 50 microliters. Actually, we usually work in a range of volume that goes from 10 to 20 microliters. And why? Why should we move from traditional in-tube samples? Why should we move from the usage of milliliters of biological samples to a few drops of blood, a few drops of urine, or a few drops of saliva? Because it's very well documented in the scientific literature nowadays that there are several advantages in terms of microsampling, and like for example the simplified process of sampling itself, the minimal invasiveness, the possibility of store these minute samples in a best way, in a more feasible way, using less room, less places and spaces in our freezers, in our fridges, in our laboratories in general. Many times these samples can be stored at room conditions, and many times these samples show a lot of advantages also in terms of sample pretreatment and analyte stability. So I want to claim immediately that I'm absolutely crazy about microsampling. So I really like working in this field. And I remember very well when James Raj came in my office many years ago, and I always stated that it was the beginning of something that was very important for my whole research group, that changed in a very significant way the way we perform analysis of illicit compounds. And virtually all types of biological samples can be transformed in micro samples. So basically, we can move from blood, from plasma, from serum, urine, to miniaturized samples of these biological matrices. And we can use fluid microsamples, but more often we use miniaturized dried samples. And the story basically starts here. The story starts with dried blood spots. That is the father of microsampling. And so basically the possibility to transfer a few drops of blood onto a special card that can be a cellulose-based card was an idea that started in the 60s for neonatal screening, and so is microsampling a new technique of these last few years? Well, no, the father of this field was born in the 60s, but actually it took a little bit of time to be exploited in other areas, and that's why in this slide I have put a date that can be the beginning of 2000, where actually several international research groups, and so the scientific international community finally decided to propose something different, different than neonatal screening and phenylketonuria screening, more in the field of drug analysis, for example. But we already knew, and we know now very well, that classic dried blood spot testing shows some limitations in terms of application of this technology to quantitative analysis. Why? Because there's a lack of accuracy due to the fact that classic dried blood spots are affected by the hematocrit, the so-called hematocrit effect. So if the hematocrit percentage changes, basically also the size and sometimes the shape of the spot can change. And so how to correlate basically the size of the spot with the volume of blood, and so finally to quantitative analysis, because when we work with the illicit compounds, it's absolutely mandatory to be able to perform a very accurate quantitative analysis. So it's not possible to use a technique that is affected by hematocrit effect. Another problem, the so-called volcano effect. Volcano effect means basically that considering a spot of blood, for example, the concentrations of the analytes, like for example drugs of abuse, the concentrations of these analytes can change going from the inner part of the spot to the peripheral part of the spot. And this is a problem. This is absolutely a problem, because if we sub-punch a spot, obviously we can pick some areas where the concentration of a compound is 10, and another area where the same analytes has a double concentration or something like that. So, well, it's possible to cope sometimes in some cases to these two big issues, the hematocrit effect and the volcano effect, but usually these are the two reasons why classic dried blood spots can represent a problem to perform for being able to perform accurate quantitative analysis. And this is the reason why the very first technology alternative to classic dried blood spots, in order to cope with the very well-known disadvantages of this technology, and volumetric absorptive microsampling is the first technology that around 2014, 2015, starting finally proposing something that can cope with these disadvantages. Volumetric absorptive microsampling is a technology based on this device called the Mitra. This device is like a pipette tip, but a very special pipette tip because it's a sampling, it's a microsampling device, able to collect a fixed and accurate volume of biological samples like blood after a finger prick, but it is not affected by volcano effect, is not affected by hematocrit effect, and so this is finally the technology able to allow people performing accurate quantitative analysis. And we had the very important chance to test this technology in our laboratory, to test the VAMS technology, emitter technology directly after finger prick, using also some automation possibilities. And the results appeared to be immediately very, very promising. That's why in my research group, together with the colleagues and co-workers of my research group, the PTA lab, the Pharmaco-Toxicological Analysis Laboratory, we have decided to shift, to move most of our analytical methods aiming at analyzing drugs of abuse, illicit compounds in biological samples to microsampling, basically. And starting from now, I'm going to propose briefly some applications that we have performed at PTA Lab in Unibo, University of Bologna. One of our papers based on the use of volumetric absorptive microsampling MITRA devices for the analysis of illicit drugs is exactly the one reported in this slide for cocaine and endogenous metabolites. Yes, we decided to test this technology, developing and validated an LC-MS instrumental analysis together. And we have decided to test the technology on blood samples and also on plasma samples. And I think that we were among the very first research groups that decided to try volumetric absorptive microsampling also to other biological samples than blood, because obviously it's evident that microsampling was born with blood for evident reason, because the sampling of blood is a usually invasive procedure, because blood is among the best biological samples to monitor substances like drugs of abuse that are psychotropic substances in a fluid, in a matrix, able to prove that a subject is under the influence of something. So it's obvious that we started from blood, but we decided also to test the technology on plasma. And this slide is just to show you that we tested the volumetric accuracy of this device. At the beginning, obviously, we trusted the company very much, but it was important to test by ourselves, because obviously each analytical method should prove to be able to grant sound analytical data. And the accuracy of these devices were amazing, absolutely. Since the beginning, it was evident how we were immediately convinced that actually it was possible to perform the analysis of cocaine and its main metabolites in blood and plasma using volumetric absorptive microsampling with no hematocrit effect. And in fact, we decided to compare immediately the results of this analysis performed on classic dried blood spots and in VAMS. And it is evident that the performance from VAMS is absolutely not affected by hematocrit effect, even when hematocrit percentage are very, very high. And what about other types of drugs of abuse? Like, for example, cathinone analogs. Cathinone analogs represent a special type of illicit compounds. Cocaine is obviously a classic illicit compound. Cathinone analogs are a family of new psychoactive substances. And so we decided also to apply this technology, to test this technology for the monitoring of very new compounds At that time, the very first cathinone analogs were really new, super new psychoactive substances. This work was published in 2016, so it was really the beginning of both the two phenomena, I would say, microsampling represented by Vance and new psychoactive substances, and cathinone analogs in particular. And we have decided to expand the testing of the application of volumetric assortive microsampling to other biological matrices, not just plasma or blood, but also urine and oral fluid. And the results were very, very promising, and the stability of these compounds were amazing. You see the stability in classical samples and the stability in Vance. It's absolutely not comparable. And then we have moved after here. We have moved to the testing and to the usage and application of microsampling, also exploiting other types of devices, like, for example, the so-called quantitative DBS. Because yes, if a device like this one, for example, is able to measure accurately the volume of blood, okay, in this case, if we use the whole spot, it's possible not to be affected by volcano effect and not to be affected by hematocrit effect. And we have used successfully also this technology of quantitative DBS for the same application some years after, volumetric assortive microsampling for the analysis still of cocaine and some metabolites. And then what about a device that is able to sample blood from a finger, so after a finger prick, but is also able to the special technology that it possesses, it's able to produce plasma spots. Yeah, this is absolutely important. This appeared immediately interesting to us, and so we have decided to test also this technology. For example, for the analysis of a special and important class of illicit compounds that are the one of fentanyls. So like, for example, fentanyl, norfentanyl, and carfentanil. Unfortunately, we know very well that these compounds represent a big problem. We have in the scientific literature data coming from plasma, and so we decided to try this device that is able to provide plasma values. And yeah, the stability is very good, the accuracy is very good, and in fact we have obtained these results that I'm showing you with pleasure, even if this is these are not published data, and we are going to submit the paper very, very soon. And then we have also the privilege of working with some experts in the field of enantioseparations, so chromatographic analysis that exploited chiral properties of some molecules. And considering that in the families of phenethylamines, tryptamines, and other cathinone analogs, there are several chiral compounds, we have decided to put together these two expertises, the expertise of my group in the field of microsampling and the expertise of the other group coming from the University of Perugia, still in Italy. And we have published, for example, this paper for the analysis of some chiral novel psychoactive substances, but exploiting microsampling. Because performing chiral analysis in this field is important to, for example, distinguish between two enantiomers of the same compound. One could even be a legal compound, for example, a prescription drug, and the other enantiomer can be an illicit compound. And there are several other reasons that proves the importance of being able sometimes, in some cases, to perform enantioseparations. but it was also very interesting to propose applications for in vitro analysis. Like in this case, this study was based on the usage of microsampling to perform in vitro metabolism of a compound that at that time, a few years ago, was really a new psychoactive substance, and there were no sufficient data in terms of metabolism. So the possibility of performing this type of analysis using microsampling was really a very challenging task, and fortunately, this study provided very important data that are now allowing several groups worldwide to perform the same type of analysis on other compounds. Obviously, all these procedures of microsampling are coupled to liquid chromatography and mass spectrometry, high resolution mass spectrometry, because we need to have a very important sensitivity, considering that we use a very minute amount of biological sample. And what about the field of anti-doping analysis? Well, we have decided to test microsampling also in this field. That is a very difficult field, because anti-doping analysis are definitely among the most difficult analyses that we can perform in a chemical laboratory. And yes, we used volumetric absorptive microsampling for anabolic androgenic steroids in blood, so in blood VAMS, with very, very good results in terms of stability also. And this is one more example of anti-doping analysis for peptide hormones and growth factors that are prohibited in competition. Even if in this case, we have decided to use microsampling for this type of analysis, exploiting VAMS technology in urine, because in blood and in urine, for anti-doping analysis, several times the problem is the stability of these compounds that decrease in terms of concentration after time. And so many times the big issue is to be able to prove the consumption of a compound after some time. And yes, also in this case, an application of microsampling for anti-doping analysis for clenbuterol, another compound that is prohibited in sports, and also in this case, a very interesting application, this time using an electrophoretic technique, or electrokinetic chromatography, a capillary electrokinetic chromatography system. And so how can I conclude? I can conclude, obviously, that the scientific literature is full, fortunately, of methods that finally are proving and proved since some years, I would say, that it's possible to perform so many analyses very successfully performing microsampling. But if we are able to develop the method on purpose, if we are able to fully validate the method following international guidelines, and obviously always take into consideration the inherent characteristics of each technology, and sometimes also the limitations that some technologies can have. Sometimes the limitations come from the peculiar biological sample that we use. Okay, we should be aware about it. And every time I present about microsampling, there's someone who comes out like, but the sensitivity is a problem. Yeah, absolutely. I've already talked about it. Yes, the sensitivity is a problem. The sensitivity can be a problem that can be solved thanks to the fact that nowadays we have very powerful technologies that many times are based on liquid chromatography and mass spectrometry. And so coupling microsampling with liquid chromatography and mass spectrometry, we can finally produce accurate data that we can really trust, and the sensitivity is not such a big issue anymore. Thank you very much for your attention, and I'm glad to listen to Michele Protti's lecture.
Okay, thank you very much, Laura, and thank you, Neil, for the opportunity to be here and to share with the whole audience our latest research carried out at the PTA lab of the University of Bologna in Italy. And I will give you an overview of our latest studies focused on the use on advanced and miniaturized sampling and analysis tools to improve cannabis testing. That is a growing need worldwide for different reasons and in different scenarios. First of all, some context on the importance of having advanced analytical approaches for cannabinoid analysis. Today, thanks to the commercial boost given by the diffusion of cannabis in many countries worldwide, we are witnessing the birth of a wide variety of cannabis-derived products able to respond to the needs of therapeutic or recreational use, and which see the evolution of the most historically classic products, such as inflorescences and concentrates, up to more advanced and refined forms of consumption, such as edibles and so on. And all these products can be further diversified according to the effects sought by users. In fact, we have more or less balanced products for both therapeutic purposes for some pathologies and for the different levels of psychoactive effects that users may seek for recreational purposes. This obviously creates a large number of possibilities for the use, which greatly complicates the task of verifying both the quality of commercialized products, but especially to monitor the use of these substances. And we must not forget that cannabis still remains the first illicit substance worldwide, making law enforcement, controls, and forensic investigations increasingly necessary. And this intersection, the bioanalytical research and new technologies are offering promising ways for the constant improvement of procedures with increasingly reliable protocols, increasingly sound results, and correct interpretation of the analytical data. And considering that this can be used in very delicate areas, ranging from therapeutic drug monitoring to forensic and anti-doping, with particular relevance in contexts involving the detection and interpretation of illicit use, and also without forgetting pharmacological and toxicological research studies that base their reliability on accurate and precise quantitative analysis. And as an example, the analysis of different biomarkers and endogenous metabolites in different biological matrices can provide different information related to the consumption of cannabis-related products due to the intrinsic characteristics of each matrix, such as the detection window. From this slide, it can be already understood how different consumption habits can provide substantially different results, making it challenging for analytical data interpretation. And let's now take a look at what happens to cannabinoids once they're inside the body. The metabolism of THC, as an example, is reported here in this slide. And THC is firstly converted to a metabolite active on the central nervous system, but whose permanence in the body is rather short, and it's rapidly converted to THC-COH, the carboxylated metabolite without any activity, and that represents the marker of choice for urine analysis. And the latter for elimination in urine is then extensively metabolized to the phase 2 metabolite conjugated with the glucuronic acid. And these two latter metabolites in particular are crucial for urinalysis, but their effective determination represent an analytical challenge. In fact, the framework of cannabinoid metabolites in biological samples following the consumption of cannabis-based products is strongly dependent on several factors, starting obviously from THC content in the consumed products, in the quantity of products consumed, obviously, the type of products and the route of the administration. For example, smoked versus edibles. The characteristics of the consumers, such as individual metabolism and body fat percentage, all contributing together to provide a complex picture that needs to be carefully evaluated and interpreted. For example, the rise of novel non-smoked cannabis products on the market presents new challenges for drug testing and forensic monitoring programs. In fact, the pharmacokinetic profiles of cannabinoid metabolites from other routes of administration can have very, very different outcomes when compared to smoked cannabis. And this represents a further complication with respect to interpretation of blood and urinary levels, windows of detection, and temporal placement of cannabis consumption. And when urine samples are subjected to hydrolysis steps, the total THC-COH concentration will include both free carboxylic acid analog and the glucuronide conjugate determined after alkaline or enzymatic hydrolysis. However, substantial inter-subject variability can happen in these patterns of excretion, and this is why more research is needed to better understand the urinary cannabinoid levels to guide interpretation of positive tests. And some studies have been focused on specific urinary markers of cannabis consumption, demonstrating how some of these are prone to degradation in classic fluid samples, even when stored under controlled conditions, in particular concerning the spontaneous hydrolysis of the phase 2 metabolite, glucuronic conjugate, resulting in THC-COH increase. Of course, this is especially crucial when the urine samples cannot be analyzed immediately, and this also poses considerable challenges in the interpretation of the analytical data to determine exactly when and how much cannabis was consumed. And at the PTA lab, we are, as you know already, we are working to give an answer to the development of innovative bioanalytical methodologies, where our focus has been on two key concepts: microsampling and miniaturization. And one of the technology we exploited is VAMS, that is increasingly recognized as a viable alternative to other dry microsampling techniques, like, for example, dried matrix spots. And here I'm directly showing you some results, because a fully validated LC-MS method was implemented together with VAM sampling and pretreatment to analyze THC together with the main endogenous metabolites. And after a comprehensive and accurate method design and development that I'm not gonna show you in detail here today, it was applied to a systematic comparative stability study between VAMS and classic fluid urine stored frozen at -20, using fortified samples and analyzed in a time frame of 6 months. On the left, you can see how cannabinoids behave in frozen fluid urine, with THC-COH increasing over time due to the spontaneous hydrolysis of the glucuronide, and a general decrease in the concentration of the other target analytes, disappearing up to 60% in a 6-month time frame, even at -20. And on the right, you see what happens in VAMS samples, simply stored at room temperature, meaning in a drawer, only protected from light and humidity. And you can see the substantial improvement in stability with analyte variation lower than 20% after 6 months. And this is probably due to the elimination of water in dried microsamples as the main substrate of chemical and enzymatic hydrolysis. Of course, these results, together with all the logistical advantages of BAM sampling, makes this technology very promising whenever the stability of analytical markers could be crucial for the final outcomes. And then we took into consideration a proof of concept application from the purely bioanalytical point of view to see if our method actually works in real world conditions. And we analyzed the urine samples from volunteers subjected to cannabis treatments and collected and dried as VAM samples. These results are in agreement with the literature data obtained through classical methodologies involving the use of high volumes of urine and classical pretreatment procedures, so thus confirming the suitability of VAMs for forensic and bioanalytical application. And other very interesting targets in cannabis testing are the so-called minor cannabinoids, and we have neutral, acidic, and barinic phytocannabinoids. Minor cannabinoids include those reported in this slide and many other ones. And many of these are considered rare, but are now emerging as new targets of selective plant breeding and are showing unique pharmacological properties. The overall actions of these minor cannabinoids often results from binding at both cannabinoid receptors and off-target receptors. So this combination makes them well suited for potential multi-target therapeutic agents for many conditions. And this means that they could become the next important compounds in the cannabis field, able to further expand both the potential benefits and at the same time their increasing presence in commercial and sometimes unregulated cannabis products makes them also relevant targets for forensic laboratories and drug control authorities. And as always happens in medicinal chemistry research, once new chemical entities are under the lens, let's say, the efforts must be focused on the development of adequate and reliable tools for bioanalysis of these emerging compounds. So, for example, being able to measure cannabinoids in biological, measure minor cannabinoids in biological samples is the key in pharmacokinetics, distribution, metabolism study, also considering the different routes of administration, as well as in monitoring real patients under therapies, including also the forensic toxicology field and post-consumption monitoring. And to support the analysis of minor cannabinoids, at the PTA lab, we adopted capillary-generated volumetric dried blood spot technology as a patient-friendly microsampling strategy. This pen-like device collects for 4 accurate replicates of 2.74 microliters from a single finger prick, offering a simple and standardized way to collect blood with minimal discomfort. Of course, to ensure reliability, we had to carry out extensive preliminary studies, including tests on sampling volumetric accuracy, optimal collection time, and the impact of hematocrit values on, for example, extraction yield and matrix effects. And the successful outcomes of these experiments validated the robustness of this technology and led us to pursue a full proof of concept application. And so following the development, optimization, and validation of the method, coupled to, of course, liquid chromatography and both medium resolution and high resolution mass spectrometry, we moved on to the application to real samples. We analyzed a set of volumetrically accurate DBS samples from users of both THC-rich products and non-psychoactive, so meaning fiber type cannabis. And the results shown here demonstrate how it was possible to identify and quantify almost all the cannabinoids taken into account for the study, apart from a couple of them, namely CBDV and CBDVA, probably due to the extremely low concentration in the starting cannabis products consumed by the subject. And so overall, these results are very promising, showing how versatile and adaptable this approach can be for a wide range of contexts, from pharmacokinetic to pharmacological and toxicological evaluation, compliance testing, and forensic caseworks. So this last glimpse on a very recent project involving a multidisciplinary team spanning Bologna, Chieti, Rome, Rimini, and Miami, and we applied FPSE, fabric phase sorptive extraction, to dried blood microsampling for the first time using functionalized fabric patches as sampling media. And FPSE follows the principles of solid phase extraction, producing clean extracts by allowing selective analytic retention and desorption, effective cleanup, and increased sensitivity. So combined with its miniaturized and dried format, we were able to demonstrate how FPSE enable simple automatable workflows that are green, stable at room temperature, and ideal for cannabinoid TDM or remote analysis. So not only the development of workflows exploiting commercial technology, but also the development of functionalized materials to expand the knowledge about the microsampling scenario, in this case applied to cannabinoid bioanalysis. And at the end of this overview, I would like to draw some conclusions and focus on some key points regarding the potential of novel advanced biosampling and analysis workflows in order to address some of the most crucial issues in cannabinoid bioanalysis. For example, VAMS microsampling and in general, advanced miniaturized technologies are able to bring several advantages in bioanalysis over classic fluid matrix assessments. In particular, they offer better sample stability, a lower risk of contamination, lower analysis costs, higher analysis feasibility, and high throughput, making these workflows particularly suitable for high throughput testing scenarios, such as forensic screening, drug compliance monitoring, and post-mortem toxicology, where stability, reliability, and traceability are of paramount importance. With this, I'm finished, and I thank you for your attention, and we are glad to take any questions. Thank you.
Thank you so much, Laura and Michele. Those really interesting presentations there. For all those attending, if you have any questions, please could you submit them through the chat function, and I can read them out to Laura and Michele. Got a couple of questions here to begin with. So, Laura, for you, the drug testing in sport area, could you just talk to us a little bit about the acceptability of this approach to WADA and other regulatory bodies, please?
Yeah. Yes, this is a very important question, I think, in this time frame, internationally speaking, because basically, I think that this is the momentum also for anti-doping analysis. In fact, just to give you an example that is significant in my opinion, the World Anti-Doping Agency, the WADA, decided to implement microsampling in official anti-doping for official anti-doping analysis. I mean that obviously anti-doping analyses are performed in urine, but also in blood, in classic ways. But the World Anti-Doping Agency decided also to allow some specific analysis by exploiting microsampling. And now there is a special working group of WADA, a special committee of WADA that works on microsampling in which way? Preparing the guidelines for those laboratories belonging to the network of WADA or not belonging into the network of WADA, all those laboratories, for example, that perform anti-doping analysis for research purposes. In this way, these guidelines can be used by everybody in a very accurate way, because, you know, Neil, at the beginning, the research groups that tried to develop something by exploiting microsampling in the field of anti-doping analysis had a hard time, actually, because it was difficult to you know, conjugate the requirements from method validation for international guidelines for method validation to all the requests and criteria of anti-doping. So it's absolutely important that finally microsampling is included in some analysis officially recognized and officially requested by sport, so by anti-doping in sports, and they are coupled to internationally recognized guidelines. This is very important. I think that the scientific community also in the field of anti-doping analysis finally recognized the importance and also in terms of future perspective for microsampling in anti-doping. It's a plus for anti-doping.
Thank you, Laura. And related to that, Michele, how do you think the acceptability of these approaches is to the legal community? Obviously, for cannabis and supporting prosecutions potentially in countries where they're not legal.
Mm-hmm.
How do you feel about the acceptance in legal systems for data generated in this way?
Okay, thank you for the question. This is a very interesting one. Of course, the acceptance from the control bodies and legal authorities is probably one of the most challenging aspects of these approaches. But as usually happens in any field, like for example in the clinical field, referring to therapeutic drug monitoring, TDM, that it's increasingly being accepted. It's the reason is that we must keep on providing reliable results, authoritative publications, bridging studies, always demonstrating the reliability with respect to classic biological fluids and with respect to decades of reference values, for example, obtained on fluid plasma. And I think if we keep on pushing on these good results on the both on the scientific community and the general public and control authorities, at some point the acceptance rates will reach that breaking point where this microsampling technologies will be considered as the new gold standard in many different fields, not only in the clinic but also in the forensic, regulatory, workplace, and so on.
Thank you, Michele. I think we've got time for one question, one further question. There's a question from Roberto here. Can you give us some insights into your future activities in this field? what do you think could be the next important developments in this field? I don't know who wants to answer that one.
I can thank you, Roberto, for the question, and I think that, for example, in this last few years, we have seen from the scientific literature that there is a lot of interest from the omics, the so-called omics, exploiting microsampling. I think that it's very important. We are working in this field as well. We have some studies running and some very recent study running. And I think that omics coupled to microsampling are very interesting. I'm looking in the monitor at Michele because we were talking about this fact in particular, and also some other new applications, like, for example, the application of microsampling to exosomes, isn't it, Michele?
Yeah, absolutely. Yeah, I totally agree. We were talking about that, but there's several new ideas looking at real world issues that make us think, okay, there's some microsampling application foreseeable for that in the future. So there's many issues that can be approached, at least with these new technologies. And I think as a research group, we are daily working on that, putting our big efforts. Yeah.
And also, just one more detail, we have discussed in a dedicated conference a few months ago some studies that are already published in the literature about the use of microsampling devices for environmental analysis. So application of microsampling to samples that are not biological samples, for example, to microsamples coming from wastewater.
Yeah,
it's not our field, but it's definitely something that is of interest.
I think there's many applications growing. I'm afraid, Alva, you submitted your question a little late there, and we've run out of time now. If you'd like to write to us, we can always submit your question about volume controlled dry plasma without volcano effect, but we don't really have time to cover that at the moment. I'd just like to thank Laura and Michele for a really interesting presentation. Just like to take a couple of moments for those of you not familiar with the patient-centric sampling interest group. If you'd like to learn more, you can use the QR code on the left-hand side to visit our website, the QR code on the right-hand side to join our LinkedIn group, which is now approaching 600 members, where we share our experiences, share publications, ask questions. A really important place to go and find out more about our community. And of course, one more slide. I didn't mean to stop doing that. One really important thing is none of what we do at the Patient-Centric Sampling Interest Group, and particularly these free webinars, would be possible without our sponsors. So I'd very much like to say thank you to our sponsors who make this all possible. Just for all of your knowledge, this webinar is free and will always remain free. And a recording of this webinar will be made available through our website in the next few days. And with that, I'd like to say thank you again to Laura and Michele, and thank you to the audience for attending today for a really interesting and important webinar. Thank you, everyone.